For farmers who are solely dependent on agriculture for living, it is important to get good yield. Traditional methods such as usage of fertilizers and manure and crop rotation are effective but not as much as modern methods. In agriculture, application of biotechnology has brought tremendous change in getting good yield, reducing maintenance cost, reducing maturing time of crops. Let's discuss various techniques of biotech which have helped farmers and agriculture industry a lot. 1. Crop selection. While growing crop, farmers have to consider required amount of water, nutrients, and time to get yield. Biotechnology has enabled farmers to find suitable crop for their farm. For example, in an area where it rains very less, crop which require less water is best suitable. And if soil contains less nitrogen, crop which requires less nitrogen is best suitable. 2. Hybridization. Hybridization is the process by which a hybrid or new variety is produced by crossing the already existing two varieties. The new variety becomes different from original ones. For example, we have different type of roses around us. Some are red in color where some are white. By crossing them, new variety of rose can be formed, which has both red as well as white pigments. 3. Polyploid breeding. In this process, chromosomal number of a plant is increased. Chromosomes are found inside the cell's nucleus and are responsible for unique traits of an organism. By increasing it, bigger fruits, thick leaves, beautiful flowers can be produced. 4. Mutation breeding. Mutation breeding is commonly used to produce traits in crops such as larger seeds, new colors, or sweeter fruits that cannot be found in nature. In this method, seeds are exposed to chemicals and radiations and are modified or mutated at cellular level. 5. Protoplast fusion. In this method, new varieties of plant are formed by merging cells of two different plants. Protoplasm is the inner cell material. When protoplasts are fused, a new cell is formed with two nuclei, and these new cells result in new type of plants. 6. Tissue culture. In this method, tissues or cells are separated from parent plant and grown in controlled environment. This method helps study the organism as well as modify it. Resultant plant differs from parent plants and its child plants also differ. 7. Genetic engineering. In this method, genes of one plant are infused in other plants. By doing so, traits of one plant can be cloned into other plant.